Now, this is also a very, very high yield image. You must know this. Must. You cannot escape it. Identify the clinical presentation displayed. Then what is the causative agent? Then other than clinical manifestation displayed, name 2 other clinical manifestations associated with the causative organism. And then choice D, I would have added, identify the best antifungal use for treatment of this. Sporotrichosis, very good. Sporotrichosis. This is sporotrichosis, very high yield. Sporotrichosis, yes. What is the causative organism? Rose Gardner's disease, very good. Sporotrix schenki, very good. Do you know why it's called Rose Gardner's? Rose Gardner's disease. Because what happens is the organism, the Sporotrix schenki that causes this disease, it lives in vegetation. So what happens is during the late 80s, they'd see many gardeners who worked in a rose field would come with such a presentation. Okay, so they'd wonder, what is going on? This presentation is known as ascending lymphangitis. Ascending lymphangitis. What that means is, if you can see this, it's clearly following the lymph nodes of this area. Okay, it follows the, not the lymph node, the lymphatic drainage of, this is, which part of the body is this? Is this the thigh or the elbow? Looks like the thigh. Can't even tell. No, it can't be the elbow. This can't be someone's biceps. This most likely looks like the thigh. So if you see this, it is following a pattern. Okay. What happens is this disease is going to follow this, the lymphatic drainage. Okay. That's why it's called Sporotrix schenki, the Rose Gardner's disease. Then clinical manifestations, other 2 clinical manifestations. And then while you're there, what is the drug of choice we use? I lost my connection. Can you please mention the high-yield question, please? So the high-yield thing was this image is very high-yield because of this pattern. It is known as ascending lymphangitis. Okay. Basically, it follows along this lymphatic drainage of this leg. Can you see? It's following that. That is known as ascending lymphangitis. Okay. Ascending lymphangitis. Yes, very good. So treatment. Then, as I said, it's usually found in vegetation. That's why it's called Rose Gardner's disease. Pulmonary lymphocutaneous, very good. Pulmonary fixed cutaneous sporotrichosis. We treat this with itraconazole. Okay, that is usually the mode of treatment, itraconazole. In fact, I've seen, I think this is the only drug that we use because we rarely mention itraconazole. It is just for Sporotrix schenki. Okay. Hope that's clear. Now, this is also another one, very high yield. So as you can see, this is a baby and they have some sort of rash at the buttocks. And this rash is not well defined. It looks irregular in shape. Okay. Can you see it? Then you're told identify the causative agent of this diaper rash. It's known as diaper rash, by the way. You can call it diaper rash, nappy rash, okay? Then you're told outline 2 mucocutaneous infection caused by the above, 2 types of specimen caused by the used in lab diagnosis, then mention 2 characteristics of the above microorganism. So what causes such an appearance? This nappy rash. Candida, very good. Now, I want you to compare this. Wait, let me stop sharing so that you see. I want you to compare this. Where is it? We'll have a good picture, but I don't have a good picture. Yes, compare that and this. Let's use this. No, let's use this. Can you compare? What is the difference between that and this? Like, how will you know in an exam this is not Candida, this is Tinea cruris? How will you know? Who wants to try? Well-defined, very good. Now, in Tinea cruris, if you were to look at the borders, they are well-defined. Okay, that's the only difference. They are well-defined borders, almost circular, regular shaped. Okay, now compare this and this in this patient. Can you see? These ones are ill-defined borders, irregular borders. Okay, now the irregular borders is Candida, the regular borders is Tinea cruris. Anytime you see diaper rash, this is Candida. Okay. Now, you've already known it's Candida. 2 mucocutaneous infection caused by the above. Oral thrush, candidiasis. Very good. Vaginal candidiasis, very good. Okay. So we have oral thrush, vaginal candidiasis, esophageal candidiasis, chronic mucocutaneous candidiasis. Then the type of specimens, we have exudates or epithelial scraping. So we usually scrape here, then we take it to the lab. Aspirates, biopsy, you can take a biopsy and take it to the lab, then definitely blood, okay? 2 characteristics of the above, so it is gram positive, It's a yeast, so it's gram-positive. Thin-walled dimorphic fungi. Okay. Then true hyphae and pseudo-hyphae. Then oval budding yeast cells. These are very specific for Candida. You see this statement? It displays both pseudo-hyphae and hyphae. Okay. The pseudo-hyphae are the elongated budding yeast cells. Okay, those are the pseudo-hyphae. And then the hyphae is the ones we saw. Okay. Are we clear? I hope you're clear. So high-end things of this, I hope you've understood it. Now, before I go to this paracoccidiomycosis, what is a dimorphic fungi? Who knows? Because I've mentioned it like 3 times and I'm yet to explain. What do we understand by the term dimorphic fungi? I give you a hint, dimorphic. So it has two forms. Yeast and molds, yes. So it exists in 2 forms. We have the yeast and the mold. And all this is dependent on temperature. Okay? All this is dependent on temperature. So at room temperature, it will be mold form. The mold form, also known as filamentous. Okay, fungus that exists in 2 forms depending on temperature. Yeast at 7 degrees, that is at the body temperature, and then mold form they at 25 degrees, which is the room temperature. Very good. So at body temperature, they are yeast. Yeast, if someone tells you a yeast, what do you understand by yeast? A yeast is a single-celled organism, usually oval in shape. Okay. How I used to remember is, so that you don't confuse these temperatures, you can say mold in the cold and yeast in the heat, like that. Mold in the cold, yeast in the heat. Mold is the filamentous form, and then yeast is the single-celled form. Mold in the cold, yeast in the heat. So it does that to survive, body temp. It's to survive and to become how can I say it? Infectious. So it will be a mold at room temperature, and then as it infects people, in our bodies we have that 7 degrees, so that's heat. It will change into a yeast. Okay, hope that's clear. Now, regarding paracoccidiomycosis, what is the causative organism? What is the route of infection? What antifungal agents are used? Name at least 2 specimens. And then what is the role of potassium hydroxide in microscope examination? Now this, if you have no clue, you will literally write nothing. Maybe you can guess here on antifungals and specimen. Okay, but yes, what do you think? Paracoccidid brasiliensis, very good. Route of infection. Now, choice E is a very general question because it can be any fungus. What is the role of potassium hydroxide in microscopic examination of Candida, crypto, histoplasma, paracoccidomycosis, all those, aspergillus, all those. Why do we use potassium hydroxide? To make it more visible, no. Inhalation of spores, very good. Now, we are 35 people and only 2 people are in the chat. Digest keratin and expose hyphae to spores, very good. Okay. Oh, that was for the people who are doing it. Now, Paracoccides brasilensis or Lutzi, inhalation of fungal conidia. Conidia are the spores from the environment. That's how we get it. And then remember, this is a systemic disease. So I told you, you use amphotericin B. Remember I had mentioned. And then specimen, we have sputum, skin scraping, infected tissue, pus, stroke discharge. And then potassium hydroxide solution digests keratin and other debris in the sample, like skin scrapings, vaginal swabs, making it easier to visualize fungal elements under the microscope. So you phrased it, you said to make it more visible. I think you should have phrased it better. For visualization, yes, that one you can get a mark. Okay. To easily visualize it. Yeah, I think that would be better. Okay. So I hope this is clear. Now, this is also another one. You already got the answer from here. Budding daughter cell, you can already tell what this is. But this is a germ tube. These are the mother cells. Then here are the budding daughter cells. What fungus is this? Yeah, you've already gotten the answer. They gave you the answer by telling you that these are the budding. Candida, very good. This is Candida albicans. What is the expected observation on potassium hydroxide? 3 systemic infections, then virulence factors of the above. So we already answered the cutaneous infections, where the vulvogenitis, oral thrush, baby diaper rash, that one. Now here they've asked systemic. Now, what happens when you have candidemia or when it disseminates? Which are the organs affected? That's how you're going to get the answers. To note the organs affected. So we have endocarditis, yes. So when it goes to the heart, it causes endocarditis, the bones causing arthritis. Okay, so I think I'd mentioned this, the budding yeast cells and the pseudohyphae. So the true hyphae and the pseudohyphae. And then candidemia, that's when it goes to blood, then it can cause urinary tract infections. GIT, pulmonary candidiasis, endocarditis, meningitis, arthritis, osteomyelitis. So think of where it goes. You see when it goes to the urinary tract, UTI, GIT, pulmonary candidiasis, when it goes to the lungs, endocarditis, the heart, meningitis, the meninges, arthritis, the bones, osteomyelitis, the bones. Okay. And then virulence factors, secretes enzymes like hydrolysis, nutritional acquisition, and degradation of host tissue. Yeast to hyphal transition. Remember, we mentioned it's dimorphic. So changes in temperature, nutritional starvation, alkaline pH causes it to transform from a mold to a yeast. Then biofilm formation, resistant to antifungals and host immune defenses. Candidilysin, lysine means to break down. Peptide toxin that promotes epithelial damage, stimulates immune activation and phagocyte. function. Okay, so germ tube is used for diagnosis of Candida albicans, usually at 37 degrees Celsius. Okay, hope that's clear. Now, before I go to this question, what do you think are the risk factors of getting Candida? What are the risk factors of getting Candida? Because they can ask that. CD4 below 50, so immunocompromised, yes, very good. Diabetes, very good. Encephalomeningitis, that is a what? Is a risk factor or a complication? Change in vaginal pH, very good. Prolonged use of antibiotics, very good. So what happens is if you if you have prolonged use of antibiotics, it's going to kill the normal flora. So what is going to happen is Candida is going to overgrow. Okay. And then is there another one? IV drug use, very good. TPN, total parenteral nutrition. Yes, good. Those are the risk factors of getting Candida. This is also another one. I've seen it many times where there's an outgrowth from the nose. It's such an appearance. Then you asked, identify the condition and most likely causative organism, appearance of such this, and then treatment of choice. Rhinosporidiosis, very good. Rhinosporidium seborrhei, very good. What do you expect rhinosporidiosis to look like? The lesion. What does this look like? To me, it looks like a strawberry. Yeah, very good. Nice. Strawberry-like appearance, okay? Then treatment. So here you cannot use an yeah, you might know what antifungal will you use to get rid of this outgrowth? You just cut, okay? We call it surgical excision. Surgical peke, very good. So it's a polypoidal febrile mass, the strawberry-like appearance containing white dots. So you can just mention what you see. Okay, then here we do surgical excision of the lesion with cauterization at the base here. So we use the laser technique where we cut here and remove it. Okay. Then number 66, mention 3 bacteria that are transmitted by the above organism. So this is a tick, as you can see, 1, 2, 3, 4, 5, 6, 7, 8. This has 8 legs. Quite a few. All the bacteria you learned from when you guys were talking about after learning spirosheets from Borrelia, all those bacteria are spread by tick, if not most. When you're learning on spirosheets, which bacteria are those? Rickettsia, good. Borrelia burgdorferi, yeah. Rickettsia connori, Anaplasma, Escherichia, Borrelia, Francisella, and Coxiella. Now, what drug of choice do we use for all these bacteria that we have mentioned? Doxycycline, very good. We use a tetracycline. This is also another one that you should know. On lab instruments, remember we had done the candle jar, extinction jar. Now this is another one. Identify the above instruments and its use. Identify the use of palladium catalyst. Then 3 bacteria that are cultured on this instrument. Fieldes, Macintosh, very good. Fieldes jar, good. Why do we add the palladium catalyst? Normally they usually ask what is modified. So the answer is usually palladium catalyst to create an aerobic environment. Okay, now let me explain how this thing works, because it's good so that in case you see it, even if they ask you, you'll know. Now, if you can see this thing, you can see there's a lid here made of metal. There's, I don't know what this is. What can we call it? The scale used to measure something here. Then there's a glass. This glass is transparent. Then inside you can see things look like Petri dishes. Okay. Now, one thing you have to know is this metal rod, the metal cover, is airtight. Okay, it's an airtight lid. Okay, then here is where you place the Petri dishes, where you have, you have the inoculated cultures. Okay, now you seal this. In the process of sealing, oxygen is going to be removed. Okay, you replace it with hydrogen. Now, this palladium catalyst that you're going to add, they're usually pellets. So usually add here, they are pellets. It's going to help. You see, when you're removing oxygen, not all the oxygen is going to, get out. So this palladium catalyst is going to help remove the extra oxygen that was left inside this jar. Okay. So remember you pumped in hydrogen. So the hydrogen is going to react with the remaining oxygen, leading to the formation of hydrogen, and oxygen gives you water, isn't it? Now, when all that happens, it creates that anaerobic environment where now the bacteria is going to grow. Okay. Then there are ways in which you can know if the oxygen is absent or some oxygen is left. There's usually a strip somewhere down here. Okay. And different colors. If the strip turns blue, oxygen is present. If the strip remains colorless, oxygen is absent. Okay. But the most important thing I want you to know is it's a Fidelis Macintosh jar. It's used to catalyze. Palladium is added to catalyze the reaction between hydrogen and oxygen to form water, removing oxygen. Then the bacteria are the anaerobes, all the Clostridia perfringens, tetani, botulinum, difficile, all those. No, difficile is not, difficile. Yeah, difficile, Bacteroides, Streptococcus, Fusobacterium, all those. Okay. I hope that's clear. Now I have a question for you guys. What antibiotics do we use for anaerobes, these ones, that are below the diaphragm. And then the next question is, what antibiotics will you use for anaerobes that are found above the diaphragm? Who knows? Mm-hmm. Any idea? Was any idea? Mm-hmm. Surely, just attempt. So let me tell you. Above the diaphragm, we use clindamycin. Below the diaphragm, we use metronidazole. Okay, that's how we treat these anaerobes. Okay. I snuck in a virus question here. What is the condition displayed and the causative agent? What is the hallmark corneal finding? And then please repeat. Repeat what? The antibiotics. Above is clindamycin. Okay. Can you explain what you mean by above diaphragm and below? Okay. Now, if you see, if you get these antibiotics, these anaerobes, let's say, for example, someone presented with a respiratory infection, and then on culture you found one of these bacteria. Let's say this one, Actinomycetes israeli, which has pneumonia features. You see that bacteria is usually found above the diaphragm. It's an anaerobe and it's found above the diaphragm. So you'll treat it with clindamycin. But now for Clostridium perfringens, which presents with, let's say, food poisoning, and then it's GI related. So that means that when you got the sample from the stool and cultured and found Clostridium perfringens, we're going to give metronidazole. So it's usually something that you should know. If you find an anaerobe above the diaphragm, so that means resp related, you give clindamycin. GI related, urinary related, yeah, those two, you give metronidazole. Okay. Yeah, so what is the hallmark corneal finding in this condition? First line antiviral, and then describe the genome and structure of the virus associated with this condition shown. What is this? Who knows? Herpes simplex keratitis. Keratitis, yes. So this is herpes simplex. Okay. This is a specific, the hallmark corneal finding. What is this? What is this called? Guys, are you guys there? Or are you tired? Let me help you guys, Basi. You guys are tired. So, herpes simplex keratoconjunctivitis, okay? And then caused by herpes simplex type 1. Hallmark is this branching. Can you see this? The branching corneal ulcers, okay? So you injected a dye and then it appeared like this. These are the branching corneal ulcers. For herpes simplex 1, we give acyclovir. Then you've been told to describe the genome. It's double-stranded DNA, okay? Double-stranded DNA, it's enveloped. Now, the whole DNA classification, there is a way that I usually classify so that I can remember. Now, this one you have to write down because it's very, very important. Now, the DNA viruses can be divided into two. We have the naked viruses and the enveloped viruses. Now, I hope you have somewhere to write because I'll need you to understand. Now, this is how I want you guys to arrange so that you don't forget. Divide that table into two. We have naked and enveloped, okay? Under the naked, the mnemonic is usually PAP, okay? Under the enveloped, the mnemonic is HHP, okay? So I hope you have divided. We have naked, PAP, enveloped, HHP. Now let's start with the naked. Under the naked, this is the order I want you to write it. So the P is parvovirus, P19. That is the first P. The A is adenovirus. The P is papilloma, which is HPV. Okay.
So can I interrupt if just you're differentiating between ssRNA and dsRNA?
No. We're not talking about RNA. We're talking about DNA viruses.
Or DNA viruses. So I have a question. There's only one ssDNA virus, right?
One single-stranded DNA virus. Yes.
It's just rota, yeah?
Yeah. No, rota? No.
No,
Rota.
not rota. Sorry.
Parvo.
No, rota is a dsRNA. Sorry, it's
Yeah.
parvo. Yeah, sorry.
Yeah. I'm going to mention things. That's why I want you to write the way I'm telling you. Then all these things that you're mentioning are going to fit in this what I'm telling you. Okay.
Wait. I'm so sorry, but do you mind like starting all over? Let me just get my pen and paper.
Okay. Let's start again. Are you guys there? Now, I don't know if I can't draw on my laptop. Now, these are DNA viruses I'm talking about. Okay. Now they're divided into two. We have naked and enveloped. Okay. Under the naked, the mnemonic is PAP. Under enveloped, the mnemonic is HHP. Okay. Now, under naked, this is how I want you to write it. This is the order. You write parvovirus. So write it down, write it on not in a straight line, write it as you're going down. Okay. Parvovirus. This is parvovirus B19. No. I've given you PAP. You're writing PPA. Don't write it like that. Write PAP. So the A is adeno. There's a reason I'm giving you this specific order. Then write papilloma. Then the last one, write polyoma. Pap. Have you guys written like that? Papilloma is HPV, then polyoma is the JC virus. Okay. Have you written PAP? P-A-P-P. Now, those are in naked. Now let's go to enveloped. Enveloped, the mnemonic is HHP. The first H is hepad DNA, which is hepatitis B. The next H is herpes. And the last P is pox virus, which is smallpox. Okay. Have you guys written it in a line? Not a vertical line, not a horizontal line. Now, so your order should be naked, parvo, adeno, papilloma, polyoma. Enveloped, hepad DNA, herpes, pox. That should be the order. Okay. Now from there, now here's where I tell you things that you have to note. At the end of the spectrum, we have, I think it would be better if I write this down. Let me stop share. Because you guys might be confused and I don't want to confuse you guys. Can you see my screen? Now this is what I wanted you to write. DNA viruses. We have naked. Then under naked, the mnemonic was. So you have parvo. Oh, I can't. It's okay. You know what I'll do? We have parvo. After parvo, we go to adeno. Then we go to papilloma. You can see my screen, right?
Yes, we can.
And then here we have enveloped. Under enveloped, we have hepad DNA. Then we have herpes. Then we have pox. Now that is the order I want you guys to use. There is a reason I've given you this order. Let me put it all on one page. Now, I'm going to remove this part, but just know that this is the naked viruses, PAP, PAP, and then here is enveloped, which is HHP. Okay. Now, parvo. Parvo here, it is the smallest virus, smallest DNA virus. It is the only one that is single-stranded. The rest, adeno, papilloma, polyoma, hepad DNA, herpes, and pox, are double-stranded. That is what is unique of parvo. Pox, it is the largest, and it is the only one that replicates in the cytoplasm. The rest replicate in the, where do they replicate? The nucleus, okay? Then another unique factor of this is it is complex. The rest are icosahedral except pox. This is the only complex. So parvo has two unique things. It is the smallest, which makes sense. It's at the beginning of this mnemonic, and pox is the largest. Parvo is the only single-stranded. The rest are double-stranded. Then pox is the only one that replicates in the cytoplasm. The rest replicate in the nucleus. How to remember that? You can say pox outside a box. Okay, the box being the nucleus. And then it is the only one that is complex shaped. The rest are icosahedral. Now, we are done there. Now, these three in the middle, polyoma, papilloma, polyoma, and hepad DNA, are all circular. The ones in the middle of the mnemonic. Circular. Circular. And then this one is circular. The rest are linear. So this is linear, 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 linear. So the last two in the mnemonic are linear. Linear, 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 linear. These three in the middle are circular. Okay. Yeah, and I'm done. Now it's up to you to, if you draw this, now any question can be asked. What is a double-stranded DNA that is linear and naked? The answer is adeno. What is a circular DNA that is double-stranded and naked? Papilloma. Why this is important is because if you get a question, you can be asked to describe its DNA genome. So when you're told to describe the genome, you have to tell us if it's DNA. And then if you're told to describe its structure, you give us the shape and whether it is enveloped or naked. Okay. Should I repeat or you guys are clear? So in summary.
Wait, before you go there, in Pavo, we don't talk about the genome being circular or linear.
What?
And also in herpes. In Pavo, yeah?
Yeah.
We don't talk about the genome being circular or linear.
Because it's single-stranded.
Sorry.
But it's linear.
And herpes is linear, yeah?
It's single-stranded linear.
Okay. And herpes and pox are both linear.
Yeah, the ones in the middle are the ones that are circular. These three. Papilloma, polio, DNA. The rest are circular. So I hope that mnemonic is clear. So in summary, you're going to say all are icosahedral except ox, which is complex. Another thing you're going to say is all are linear except papilloma, the ones in the middle, papilloma, polyoma, and bad DNA, which is hepatitis B, which are circular. And then the last one is all are double-stranded DNA except Pavo, which is the only single-stranded DNA. Hope that's clear. Let me copy paste this. So this is something you have to know because it forms a basis of answering questions. Okay. Now, in terms of shape, genome, structure, you should not have an issue with DNA viruses. Clear? Any question there?
Is there such a mnemonic for RNA?
Nah, not that I know of. RNA, the issue about RNA, there are so many. Maybe
Okay,
a few.
I think I'll find
Mention
one
a few.
then.
Yeah.
I've seen one, but I'll send it to you, and I'll also send these guys. But I don't know if it'll help them because, like you see, you first of all with RNA, you first divide it into dsRNA and ssRNA. In dsRNA, there's just one, which is the rota, right?
Yeah.
And then in SS, it is further subdivided into positive sense and negative sense. Yeah, so like in the positive sense, naked icosahedral is going to be this mnemonic called pen. So it stands for, the P is for is for Picornaviridae, and then the E is for Hep E, and N is for Neuro. So like that. I think I'll send them table. It's fine.
Yeah, you can send them
Thank
a
you.
table. That would be good. But I hope the DNA viruses are clear now.
Yeah,
One,
this made it so much simpler.
yeah, it's easier because no need for cramming. You know, sometimes you'd be like, oh, what is it? Because in exam, they can bring you a picture and then you're told maybe to describe adenovirus. Then you'll be like, oh my God, I just remember it is double-stranded. I mean, I can't remember anything else. But if you draw that table like that, you can, from this description, you can mention any virus and you give the characteristics. Also orals, whether that is something that can help you during orals. Because a lecturer can just ask, hepatitis B, please.
But also these viruses,
Yeah.
you see these viruses, they're also subdivided. Like herpes, it's these herpes 1
Yeah.
to 8, and then pox
Yeah.
again, it's divided into smallpox, monkeypox, molluscum contagiosum.
Yeah. But at least I've given you a basis. So you can like
Wait, so if you want to remember the others, you'll just have to like kind of cram them, right?
unless there's a mnemonic. I'm sure you guys have
I'll
better
I'll I'll try. I'll look for it. It's okay. Thank you though.
now. So I hope that's clear. We're left with 4 more questions, then we're done.